In this study, we are looking at how the brain learns from rewards. We study this by asking volunteers to play games in which they can win as much money as possible, and we record their brain activity while they are doing this.
Is it comfortable?
It's not too bad, actually.
And why do we look at the brain? Because we know that dopamine neurons are really critical when we anticipate rewards. They fire when we see something good is about to happen, and they stop firing when we don't get that good thing. But what we show in this project is that this is not the only signal that our brain elicits for rewards, but actually this is a two-stage process where we have an early salient signal that tells us how important things are, followed by an evaluative signal of how good and bad that thing is. So in fact, when we process rewards, we start processing them much earlier than we thought before.
So imagine, walking past the coffee machine at work and you hear that loud sound of the machine finishing brewing a coffee, and then ultimately that gives you that level of arousal that when you go back to get your own cuppa, you ultimately feel like that's more energizing and more exciting than it really is, even though it's just that same average, mediocre blend of coffee that you have every day.
This project is relevant to understanding mental health disorders where we know that the reward system is disrupted. And addiction is a great example. We know that the reward system is biased in addiction. And what I think is that it might actually not be that dopaminergic evaluative response, but the earlier salient signal that makes people find things more important than they actually are. So this is definitely something that we want to pursue, and in the long run, hopefully to design treatments that might be more tailored depending on these individual mechanisms at play.
So ultimately, we're using two different neuroimaging modalities here. One is the EEG that we collect from electrodes on the surface of the scalp, and it gives us very good temporal precision. So in other words, it tells us how different events in the brain unfold in time.
And this is combined with fMRI at 7 Tesla, which is very good to localize in the brain where do the things happen.
So there is something to be said about putting the two things together, measure them simultaneously, and ultimately leverage what's good about each of these imaging modalities and have both high temporal as well as spatial precision of the cognitive problem that you're studying.
To our knowledge, only very few sites are able to do simultaneous EEG fMRI at 7 Tesla because the technology is not really available. There are not commercially available options. And here we are very lucky to have in the Imaging Center of Excellence the Coil Lab, led by Shajan, who designed and developed this coil specifically for our interests. And it can also pave the way for future studies on mental health disorders. We are trying to do some developments to allow that using a more patient-friendly coil.
The current industry standard head coils are really claustrophobic and they are very close-faced. It's a big window on the top so that the patients can see through, and the mirror system will allow them to see the operators when they are getting scanned. So it's not just about making a hole on the plastic. It's a lot of electromagnetic redesign and simulations.
We can actually have a person with the cap, the EEG cap on, and we can connect the cap to all the EEG equipment, which are going to transmit the signal to our computers where we record it from. So this design is really critical to do simultaneous EEG fMRI at 7 Tesla in a safe way. And we also minimize the artifacts from the scanner from having very short cables between the cap and these equipments. And this, as you can see, it's impossible to do with these commercial coils.
Ultimately, I see this as an opportunity to introduce a new tool for cognitive neuroscience and for other labs and other sites to adopt going forward.
The most exciting finding from this project is that it shows very clear results that are very consistent with what other people saw in other animals, such as monkeys and rats. And this translation from other species to humans is quite rare. So we are feeling like we are really bridging this gap between basic neuroimaging and human cognition and neuroscience. we are understanding the basic neural mechanisms behind behavior. And ultimately, in the long run, what we aim is to be able, with this fine-grained precision, to detect when things go wrong in the brain, and then we can design interventions that target these specific processes of interest. If people want to find more about our research, please click on the link to our website.